Hello guys, in this video we will be learning about the venous development. We will be learning this venous development in parts. This video is part 1 which will be dealing with the visceral vein development, namely the portal and hepatic venous system. In our last lecture, we have studied about the sinus venosus development and how three paired veins are contributing to the development of sinus venosus. As you can see here, the blue structure is the sinus venosus and we have the green structure. The cranial green structure is the anterior cardinal vein, and the one which is caudal is the posterior cardinal vein, and both of this join together to form what is called as common cardinal vein, and this common cardinal vein drains into the sinus venosus. You also see two other paired venous structure, namely the yellow as well as the pink colored venous system. So the pink one is the umbilical vein and the yellow is the vitelline vein. So in this picture you could see both the umbilical and the vitelline vein are draining certain structures. Since the topic is about the visceral veins, we are not dealing about the cardinal system which is in green color. So you could see the pink one is draining a red structure here. So the pink one here is the umbilical vein and the red structure is the placenta. So umbilical vein is draining placenta, and you could see the yellow structure which is the vitelline vein which is draining this orangish colored structure which is nothing but yolk sac. Now let us study the changes that are happening in the vitelline veins. The picture on the left shows the entire venous tributaries including the sinus venosus, cardinal system, vitelline veins as well as umbilical veins. So for study purpose, I have removed the green cardinal veins, the pink umbilical vein as well as placenta. So the picture on the right we have includes sinus venosus, vitelline veins as well as yolk sac. So for study purpose, I have changed the orientation of the vitelline veins, and what we are looking is just the middle as well as the distal portion of the vitelline veins as well as the yolk sac. In the last picture here, we could see an additional change that is happening in the form of tubular structure arising from the yolk sac. What is this tubular structure? This tubular structure is nothing but it is the formation of the midgut, and it forms out of the endodermal cells of the yolk sac, which will later form the duodenum. Therefore, apart from the yolk sac, the vitelline vein also drains the midgut. Therefore, the vitelline venous system are sometimes known as omphallomesenteric venous system. Here, in the last picture, we could appreciate the first change that is happening in the vitelline venous system. You could see some anastomotic sites across the two vitelline veins. which is wrapping the midgut. So the important point here is that the distalmost points of the vitelline veins are anastomosing in an anterior fashion, or in other words ventrally, whereas the portion which is proximal to the caudalmost portion are anastomosing in posterior fashion. In other words, they are anastomosing dorsally. So far, we were discussing about the middle as well as the distal portion of the vitelline venous system. Now we will look into a larger picture which also includes the proximal portion of the vitelline veins. So this was the middle and the distal portion, and above this we have the proximal portion of the vitelline vein. As we know, the caudalmost portion getting anastomosed in a ventral fashion, and the portion which is just above it or proximal to it is getting anastomosed in dorsal fashion, thereby wrapping the developing midgut. Now, the proximal portion of the vitelline vein is also getting anastomosed, and this anastomosis is again in ventral fashion or anteriorly to the midgut. So, in total, we have two anterior anastomosis and one posterior anastomosis across the two vitelline veins. The importance of the proximal portion of the vitelline venous system is that it will be affected by one growing structure, which will be discussed later. Let us move on to the changes that are happening in the umbilical vein system. So once again, I've just removed the green colored cardinal system and the yellow colored vitelline vein system. So all we will be discussing here is the sinus venosus along with the umbilical venous system. So for study purpose, I've just made this orientation a little different. So the change that is happening in the umbilical vein system is that, which will be taken as change number 2. You could see the proximal portion of the umbilical vein system, which is draining into the sinus venosus, is getting regressed. So as we know, this is the placenta and these are the right umbilical vein and left umbilical vein. So they are carrying the oxygenated blood from the placenta and they drain into the sinus venosus. But few changes happen in the proximal portion of the umbilical vein, thereby they are getting regressed. So this is the number 2 change that is happening in the visceral veins. Now we will combine both the vitelline vein as well as the umbilical vein. As we saw before, the number 1 change that was happening was the formation of the anastomotic channels in the vitelline veins, and change number 2 is the regression of the proximal portion of the umbilical vein that is draining into the sinus venosus. So these two changes are interdependent. Let us see how it works. Here we could see in picture 1 the sinus venosus along with the vitelline vein and the yolk sac. And picture number 2 we could see the sinus venosus along with the umbilical vein system. So the number 1 change that happened is the anastomosis formation. The number 2 change that has happened is the regression of the proximal umbilical vein. Here we also see a number 3 change that has happened. So this number 3 change is nothing but it is the change in the form of yolk sac regression. So the yolk sac starts regressing. So these are the 3 changes that are happening. The first picture shows the combined vitelline vein system and umbilical vein system. Since the proximal portion of the umbilical vein are getting regressed, now the umbilical vein has to find its way to deload the oxygenated blood from the placenta. So it finds a way by creating an anastomosis with that of the vitelline vein. So this blue colored structure is the anastomotic channel that is developing between the umbilical vein and the vitelline vein. Now, whatever blood that umbilical vein is carrying from the placenta is now getting channelized to the vitelline vein through this anastomotic channel. So, the anastomotic channel as well as the portion of the vitelline vein across the anastomotic channel and proximal to it gets mightier. Here you could see it is enlarged. The blue one is the vitelline vein along with the anastomotic channel. And you could see on either end of this, the pink one, it is the regressed form of the proximal portion of the umbilical vein. So the fourth change that has happened is the formation of the anastomosis across the umbilical vein and vitelline vein and the enlargement of this portion along with the vitelline vein. So all we have left out is the vitelline vein system along with the anastomosis, and now it is termed with a newer term called as hepatocardiac vein. So on the right we have the right hepatocardiac vein. This is called as the hepatocardiac vein. So right hepatocardiac vein. Here we have the left hepatocardiac vein. In the previous slide, I had discussed that the proximal portion of the vitelline vein will be influenced by a growing structure. We know that septum transversum forms the diaphragm and it separates the heart from structures below. You also know that septum transversum gives rise to liver buds. So the growing liver engulfs the structures that are present across it and it influences these structures. So the proximal portion of the vitelline vein as well as the umbilical vein will be influenced now by the growing liver. Now all that we have is the right and the left hepatocardiac veins because the proximal umbilical vein has regressed and formed a anastomotic channel across the vitelline vein. So here we have the growing liver. The last picture shows the cut section across the growing liver. So the channel that is getting influence is the right and the left hepatocardiac channel, as well as the proximal anastomotic channel across this vitelline vein. Let us move further. The fifth change that is happening across the system is that the left hepatocardiac venous channel is getting regressed. Here you could see it has got regressed. And the sixth change that has happened is the right umbilical vein here is also getting regressed. So, because of these changes, we understand that whatever blood that is getting exchanged from the placenta is only through the left umbilical vein. So the left umbilical vein, which carries the oxygenated blood, moves forward, but it cannot empty its content into the left hepatocardiac vein because it has already got regressed. Hence, the left umbilical vein has to find its way to empty its content into the sinus venosus. Therefore, now an anastomotic channel forms between the left umbilical vein as well as the right hepatocardiac vein. So the green color structure over here is the anastomotic channel across the left umbilical vein and the right hepatocardiac vein. This is the seventh change that is happening across the visceral venous system. This anastomotic channel is named as ductus venosus. Now let us look into the eighth change that is happening within the system. So the eighth change is the formation of numerous anastomosis. So this is happening across the proximal anastomotic channel within the vitelline venous system. So you could see in this picture the proximal portion of the vitelline vein has a ventral anastomosis, and as I told you before, it will be influenced by the growing liver. As the liver grows, it invaginates more anastomotic channels, thereby breaking down the proximal ventral anastomotic channel across the vitelline vein. It forms structures known as hepatic sinusoids. So the eighth change that is happening is the breakdown of the anastomotic channel into numerous channels named as hepatic sinusoids. Now, what is the ninth change that is happening? The ninth change that is happening across the middle and the distal portion of the vitelline vein. Some of the anastomotic channel gets disappeared and some portion of the vitelline vein also gets disappeared. Anastomotic channel that is getting disappeared is the caudal most ventral anastomotic channel, The other vitelline vein parts that is getting disappeared is on the left, the part of the vitelline vein between proximal and the middle anastomosis. Again on the left, the portion of the vitelline vein below the middle anastomosis. On the right, the part of the vitelline vein below the middle anastomosis. So these are the parts which get disappeared. So the remaining will be the proximal as well as the middle anastomotic channel and the portion of the vitelline vein between the proximal and the middle anastomosis. In the previous slide, we had discussed about the disappearing anastomotic channel as well as some parts of the vitelline vein. Now, the 10th change that is happening across the visceral venous system is the formation of the portal vein. So, here you have a structure, here you have another structure. So, this is the splenic vein and this is the superior mesenteric vein. Both these veins independently grow and they join at the confluence of the middle anastomotic site and they drain into the middle anastomotic site to form what is called as the portal vein. In the last picture, you could see the rotation of the gut, namely the duodenum, and how this is the spleen, this is the splenic vein, this is the superior mesenteric vein. They are draining into the middle anastomotic channel which is placed posteriorly or dorsally to that of the duodenum and they form what is called as the portal vein and this portal vein is draining into the liver. So it drains into the liver in such a way this part or the part of the vitelline vein between the proximal and the middle anastomotic channel forms the right branch of portal vein and the proximal anastomotic channel itself forms the left branch of portal vein. Let us revise all the changes that are happening within the umbilicovitelline vein system. The first change that is happening is within the vitelline vein is the formation of the anastomotic channels between the two vitelline vein. The cranial and the caudalmost channel are going to be ventrally or anteriorly placed and the middle portion is dorsally or posteriorly placed. The second change that is happening is in the umbilical venous system where the proximal draining end of the umbilical venous system into the sinus venosus is getting regressed. And the third change is happening within the yolk sac where the yolk sac is getting regressed and starts disappearing. The fourth change that is happening, because the umbilical venous system, the proximal end is now getting regressed, it has to drain some way. So it starts forming anastomosis with that of the vitelline venous system at its proximal end. So thereby it forms a channel which is now called as the right and the left hepatocardiac venous system. So whatever the changes that are happening are getting influenced by the formation of the lung buds. The developing lung buds will influence mainly the proximal portion of the umbilical and the vitelline venous system. The fifth change that is happening within the left hepatocardiac vein. The change that is happening is the regression of the left hepatocardiac vein. The sixth change is happening within the right umbilical vein. So, right umbilical vein gets regressed and starts disappearing. The seventh change that is happening here is the formation of the anastomotic channel, namely the ductus venosus. This happens because the regression of the left hepatocardiac vein, therefore the left-sided umbilical vein must find a path to drain into the sinus venosus. So, starts forming a anastomotic channel between the portion that is just below or distal to the regressed left hepatocardiac vein and the right hepatocardiac vein. So, this is called as the ductus venosus. The eighth change that is happening between the anastomotic channels that are present, mainly the cranially placed anastomotic channel of the vitelline vein. So, it gets breakdown into multiple venous sinusoids because of the development of the lung buds. The ninth change again happens in the vitelline vein. Some portion of the vitelline vein gets regressed and some anastomotic channels also regressed, thereby leaving only few. The 10th change that is happening is the connection or the drainage of the independently developed splenic vein as well as the superior mesenteric vein into the leftover part of the vitelline venous system, thereby forming the portal vein. And this portal vein gets drained into the liver. The last picture shows the adult form of venous system. So here you have the liver. These are the sinusoids. This is the right hepatic branch, left hepatic branch, and you have the common hepatic vein. This common hepatic vein drains into the structure called as IVC. Here you have the superior mesenteric vein, splenic vein, forming the portal vein, and portal vein draining into the liver. This is its right and the left branch. With this, we have completed the development of hepatic and portal visceral venous system.